One of your learning objectives for Lab 2 is to examine and recognize the features of compact and spongy bone. In the lab, you'll be looking at a three-dimensional model of compact bone and histology slides of both compact and spongy bone. In this tutorial, we'll take a look at compact bone. You may also see this type of bone called dense bone or cortical bone. The basic functional and structural unit of compact bone is the osteon. And sometimes you'll also see the osteon referred to as a Haversian system. And I'll highlight here one individual osteon. In this scanning electron micrograph, you can see that the osteon is a three-dimensional cylindrical structure. At the center of each osteon is the central canal, which I'll highlight here. This is the central canal. The central canal is a channel that houses blood vessels and nerves that supply the bone. Surrounding the central canal, the bulk of each osteon is composed of concentric rings of bone tissue called concentric lamellae. And I'll show them here in blue. So these rings are the concentric lamellae. In the boundaries between the concentric lamellae, we find small spaces called lacunae. And I'll circle a few of them in red. Cells called osteocytes reside within the lacunae. Osteocytes are mature bone cells derived from osteoblasts that have become trapped in the matrix that they've secreted. Small channels extend between the concentric lamellae, and those channels are called canaliculi. The osteocytes extend small cytoplasmic projections into the canaliculi, and this permits intercellular communication between the osteocytes. The canaliculi also connect to the central canal so that the blood vessels in the central canal can exchange nutrients, waste, minerals, gases with the osteocytes via the canaliculi. So let's take a look at the histology of compact bone as you're going to view it under the light microscope in the lab. This slide shows a high power magnification of bone tissue, and it looks a lot like what you'll see under the microscopes in the lab. So we'll highlight some of the features of this tissue. First, we have the individual osteons, and in the center of each osteon, the central canal, and the lamellae, which are the concentric rings of tissue that make up the osteon. And between each of the lamellae, we have lacunae, which are the spaces that house the osteocytes. And within the lacunae, we have the osteocytes. What we didn't see in the scanning electron micrograph and what we can see in this light micrograph are these small channels between the lamellae, the canaliculi. These are spelled canaliculi. And again, the osteocytes extend cytoplasmic processes into the canaliculi, which enables them to communicate with each other and with the central canal. So those are the most important features of the osteon. And next we're going to switch to a lower power and highlight a few structures that are in the bone tissue but not part of the osteon proper. In this low power image of bone tissue, we can see many osteons, which are highlighted here. And then in between the osteons, we find what's called interstitial lamellae. The interstitial lamellae are remnants of osteons that have been partially reabsorbed. Another notable feature we can see here are these perforating canals, which I've highlighted. The perforating canals are also called Volkmann's canals, or Volkmann canals. And these canals run perpendicular to the central canals, and they connect multiple central canals. You can see that sort of right here, that these two central canals are connected by a perforating canal. The perforating canals also house blood vessels and nerves, and they form a vascular and nervous network between the osteons. In lab this week, be sure to view the histology slides of both compact and spongy bone. This concludes this tutorial.